Okay, awesome. Great. After the smart people have solved the technical issue, it's my turn to apologize for the delay and to introduce to everyone our first speaker of the series of lectures that we have during this NGSprint hackathon. And it's my great pleasure to introduce Judith Zaugg, who is a group leader at EMBL and Molecular Medicine Partnership Unit. Judith, she did her PhD at EMBL, but at EBI in Cambridge, and there she was working on integration of different omics datasets in yeast, actually, and she uncovered several non-canonical gene regulatory mechanisms in yeast. And later she joined as a postdoc to Stanford University, and it's there that she started studying variability of different regulatory mechanisms across individuals and linking them to their genetic variation. And she actually published landmark studies that established a genetic basis of epigenetic markers, and that, among other things, exemplified the importance of enhancers in various disease mechanisms. Now, Judith, she leads her own research group at the EMBL, and they're continuing this work on the genetic determinants of the expression variation. And they are developing computational tools and approaches for integration of multi-omics data and all this to study regulatory mechanisms with the aim of better understanding the molecular basis of complex hereditary traits and different genetic diseases, particularly focusing on neurodegenerative and hematopoietic diseases. I don't know about you, I'm very excited to listen to this talk, and Judith, you may now go ahead.
Thank you very much. Thanks for this very kind introduction, and also thank you for the invitation to speak here at your summer school. So I'm going to give you a bit of an overview of how I think about epigenetics or systems epigenetics and how I think we can actually use this. And data integration is kind of a big part of systems epigenetics, and how we can use this as tools to study disease mechanisms. So before I jump into can you actually see my mouse? It's always a question.
Yes, we do.
Yes, okay, great. Okay, so before I jump into the topic, let me just introduce you a little bit of what I think is one of the key underlying motivations of the research that we do in the group. And one of the questions that kind of underlies many of the studies we're doing is to understand what makes each individual, and you can extend that to patients also, what makes each individual or each patient unique. And then the second question that follows immediately is how can we then use this variation to actually understand basic biology or disease mechanisms? And people vary in many different ways. They vary in how their faces look like. They vary in their propensity of getting a certain disease when they're sick. They vary in their sizes. They vary in how they respond to drugs. And also, of course, patients vary in terms of the mutations that their cancer has. So there's lots of variation that we have among the population. And if we break this down, I think if you look at the effects on phenotype, on the factors that drive the variation on phenotype, this is multifactorial. And this is mostly driven by two big factors. One is the genetics part and one is the environmental part. And when we look at the genetics part that drives variation, I'm talking now specifically about human individuals, then we have made the field has made tremendous progress in the past 20 years or so, when we have started to associate the single nucleotide polymorphisms, which are mutations or not mutations, variants in the germline genome that are like a one base mutations, and that these SNPs, how we call them, have been associated through genome-wide association studies to hundreds of thousands of diseases and traits. When we think about the environment, we have made tremendous progress in understanding kind of how the big fluctuations in the environment, like toxic air and all kinds of pollutions or sunshine or whatever, has an impact on phenotype. Also, we have made a lot of progress in understanding, that nutrition or our kind of immediate microenvironment of what we put into and onto our bodies can affect phenotype. And of course, there's tremendous studies going on at the moment in the kind of within body communication of cells or microbes or whatever things we have inside of our bodies. And when we now think about and many of these mechanisms have been linked to some extent to some epigenetics in a broad sense. Now, what are the key challenges that we have in those two fields? On the one hand, when we look at the genetics, I think the biggest challenge here is really a lack of interpretation, because for this, we have lots of these associations, but for 95% of these non-coding of these SNPs, they lie in the non-coding regulatory elements. So they don't lie in protein coding genes, which would make it kind of a little bit easier to understand what could be the potential mechanism of how they act. But most of them actually lie in non-coding elements. So we still don't know what this means. So I'm thinking here the key challenge is a lack of interpretation of these variants. Whereas for the environmental challenges, I think here we not only have a lack of interpretation, but actually a lack of data. So we still have no idea how these kind of big environmental factors actually influence epigenetics and how these epigenetics then influences complex traits. So what does this mean in kind of in a real world? So I think when we think about precision medicine in an ideal world, what we would say is we would take the genetics of an individual once that person gets sick, and we would get kind of the environmental factors that this person has been exposed to, and we would calculate the lifetime risk, and we would calculate kind of the epigenetic exposures, calculate the cumulative risk, and then understand the exact current molecular state of that person once he or she becomes sick, and then we propose a treatment. Now, of course, we all know that this is not possible. This is not feasible at the moment, and partially this is because of these real-world challenges that we still having trouble of interpreting the data, and we actually lack a lot of data of the environmental parts. Now, of course, what precision medicine does do, or personalized medicine, is that it takes biomarkers, all kinds of biomarkers, to inform current risk and also inform treatment. And I think this is already tremendously better than not doing this. So I'm not saying that personalized medicine is not doing anything. I'm just saying that it's mostly based on biomarkers. And what I think the field of systems epigenetics here has to offer is to try to understand kind of the underlying mechanisms that lead to these biomarkers, for example, through quantitative trait loci analysis, but also understanding signaling pathways, chromatin regulation, and also gene regulation that can lead to these changes in biomarkers, which would then bring us from only informing current risk and treatment to actually understand the cause potentially and potentially propose new treatments. So this brings me to the vision that we have in our group, which is to understand this interplay of epigenetics, environment, and at the molecular level, and kind of by this push personalized health into the next frontier. So what I'm showing you here is that, of course, instead of measuring only the DNA and then associate it directly to the complex trait, you can measure all these intermediate molecular phenotypes like chromatin, RNA, protein, metabolites, and so on. And what we are doing in our group, or what we are kind of aiming for, is to do this molecular profiling across many individuals and then generate what I call disease and cell type specific regulatory network models, where we essentially connect the signaling parts with transcription factors, with gene regulatory elements and target genes, which would then allow us to actually integrate the different parts that I just explained to you, the genetics and the environmental influences, and that would then allow us to get this patient a cell type specific response to perturbations, right? So we would know if somebody gets perturbed, we would get this kind of response in one patient, but with a different genetic setup, it would be a different response, for example. Then, of course, we can use these models to again refine our kind of cell type specific regulatory network models. Now, this is the big vision that we have in the lab. And what I want to first start with in this talk is to tell you how we build these regulatory network models, and how we then, in the second part, I will then show you how we apply them. So in my opinion, there's 4 building blocks that we need for this regulatory for these regulatory network models. On the one hand, we need to define what are these regulatory elements. This can be enhancers and promoters. Then we need to be able to measure transcription factor activities. Then we need to be able to connect these transcription factors to their genes. And finally, we need to be able to connect transcription factors to their upstream signaling pathways. So to start with the first part about defining the regulatory elements, here we're taking a very simplistic approach. Essentially, we define regulatory elements as genomic positions that regulate transcription, such as promoters that are close to transcription start sites, or enhancers that are further away. And we define them essentially by these chemical modifications of histone tails, in other words, chromatin marks, that have been mapped actually across many cell types, and they have been associated with activity or inactivity, different chromatin marks that are kind of marking an active cell or an inactive cell. And what has been done a lot, actually, this is the ENCODE framework has really pushed the boundaries on this one. So the third round of ENCODE has really provided a tremendous dictionary of how these regulatory elements vary across cell types. Of course, what is a little bit more difficult, and this is where our research goes towards, is to understand the subtle differences that we find within the same cell type, but across different individuals. So here we only expect like small differences. You can see here, maybe one enhancer is a little bit less active and then the gene is a little bit less expressed, but it is still there, the baseline expression. Whereas if you look at cross cell types, of course, it is very much different. So this is this was the paradigm shift also that was mentioned in the introduction, where we actually found many of these genetic elements, many of these so-called epigenetic chromatin marks that we have here. These are 3 different marks, actually are correlated with the genotype. So there seems to be an underlying genetic signal. And we also found that the transcription factors mediate many of these different, epigenetic signals. So essentially, you have a SNP that don't allow that prevents the transcription factor from binding, that prevents him from recruiting histone-modifying enzymes, and you end up with like a less open chromatin here, whereas if you end up with an open chromatin here, whereas if you can't have the binding, you don't have the chromatin open and you don't get the gene expression. This is a very simplistic way, but just to show you that it's also not super simple to define what is now epigenetic and what is genetic. That's why I like to look at it in a very global way that kind of integrates both signals at the same time. So essentially, we define these regulatory elements using chromatin marks. So the next step that we have to do is to measure transcription factor activities. And here I have to explain you a little bit that transcription factors are actually very challenging proteins to understand kind of their activity, because first of all, they're very lowly abundant. So you typically don't observe them actually in proteomics data. I'm just showing you here a figure. I'm gonna go to this later on actually again, but just to show you, genes detected by different layers, for example, by protein, by RNA, by chromatin. The ones that we can detect on proteomic studies with mass spectrometry actually is pretty much depleted of transcription factors. And also they are very heavily post-translationally regulated, which means that if we just look at the RNA level, we actually won't get the full picture of how active a transcription factor actually is. So on the other hand, what we are very good at doing with transcription factors is we are very good at predicting their binding sites in the genome, and we can very easily measure chromatin modifications. And so from this kind of simple concepts, we have developed this approach to combine transcription factor binding site predictions with chromatin signal to kind of identify an aggregate measure of transcription factor activity. For example, here we have a lot of chromatin openness around the binding sites. Here we don't have that, so we just kind of infer that in this population transcription factor activity was high and here it was low. So we have formalized this relationship in a tool that we call DiffTF, or the Differential Transcription Factor Activity, that essentially uses some kind of chromatin activity data, transcription factor binding sites, and then can infer differential transcription factor activity. We have applied this in many papers, so you're welcome to have a look at this. But I think that the essential part here is that we have now a way of defining transcription factor activity from chromatin marks. And we can also identify regulatory elements from chromatin marks. So so chromatin can serve as a readout for both, which makes it easy to connect these 2 layers. So just again, to go back, we have now a very nice tool to look at transcription factor activity. Now the third part is I think the most challenging one, because now we wanted to connect the transcription factors to genes. And of course, there's a huge literature actually on all kinds of co-expression, codependency, or TF knockdown networks that have correlated or somehow associated transcription factors to their target genes. However, what we are really interested in is this SNPs that I've told you about, this disease-associated genetic variants that mostly lie in non-coding regions. So you can't just connect transcription factor to target genes. We actually want to know the enhancers that mediate this interaction. So for this, we kind of split this problem into 2 problems. We have, on the one hand, transcription factor to regulatory element connections here, and on the other hand, the regulatory element to target gene connections. And the way we're doing this is simply with a covariation approach. We look at the expression across individuals. Now, this is important across individuals because this gives us really this subtle variation that we have across the population within the same cell types. And then we use a false discovery approach by just assuming the foreground is the peaks that have a transcription factor binding site, the background is the peaks that don't have a binding site, and we can then calculate, a kind of a false discovery rate of how likely the transcription factor is actually connected to the chromatin marks. A very similar approach we can then use from chromatin marks to the target gene expression. Just here, we also use in addition physical proximity to make the connection. Now, what is very interesting actually, when you look at this, I'm showing you here, we'll take correlations that are greater than 0 and also correlations that are lower than 0. And what we can do actually with this is, if we think about it, some transcription factors can act as activators. So we would generally expect that increased expression would lead to increased chromatin accessibility for the activator. Whereas for repressors, if a transcription factor would be actually a transcription repressor, we would maybe expect the opposite pattern. And this is kind of showcased here on one example where we look at the specific footprints of chromatin accessibility of a typical activator, where we find indeed a footprint of very open chromatin surrounding the transcription factor binding site. Whereas for repressors, we find that even though there is a little bit of accessibility just on top of the binding site, the chromatin surrounding the bigger region of this binding site is actually repressed or is like inactive. So this kind of indicates that we can indeed use this as a way to identify activating and repressing TFs. And this was quite interesting actually on its own, because when you look at the literature, and I'm just taking here the classifications from one database and how often a transcription factor has been classified as a transcription repressor versus activator in primary literature, you find that most transcription factors are actually kind of equally often found as being repressing and activating. So we thought we had to validate this thing and this kind of theory, because we couldn't validate it with the literature essentially, because each transcription factor was multiple times annotated as one or the other. So we used a cohort data, great data set from Christoph Bock's group in Vienna to classify transcription factors into these activators and repressors based on this network that I just told you. And then we treated an independent cohort with a specific drug, ibutrenib, in this case, and compare transcription factor activity and expression. And the expectation was that for transcriptional activators, we would find a positive association between the TF expression and its activity on the chromatin. And for the transcription repressors, we would find a negative association. And this is indeed what we find. So for the activators, we find a positive association. For the repressors, we find a negative association. So we think this is really a way to globally find whether a transcription factor is now acting more as a repressor or as an activator. And I think this is very important if you want to understand the downstream effects of, for example, say inhibiting a transcription factor. So what we have to do now, when I talk again about our gene regulatory network model, we have to update it and actually specifically say whether transcription factor is now having an activating or repressing action. And when we did this analysis across many cell types by now, we generated these gene regulatory networks. And I'm just showing you here a bunch of transcription factors that actually change their mode of action across different tissues or across different cell types. So I think it's actually very interesting to think about this, that transcription factors do change their role. And of course, there's many ways of how they could change their role. They could have different regulatory complexes, so they could cooperate with other transcription factors. But I think the take-home message that I want to focus on here is that really depends on the context of whether a specific transcription factor is now acting as a repressor or as an activator. Again, kind of highlighting that we need these cell type specific networks if you want to really understand these details. So what I've shown you here in this building block, we can now link transcription factors to enhancers based on covariation across individuals. And we have actually also written a nice R package. So in case you want to try this out, this regulatory network analysis, and you have data sets of RNA-seq and ATAC-seq, for example, across multiple individuals, this is the software that we have developed for it. So now let me go to one example to show you how we can now integrate, how I think now we can use chromatin to integrate kind of these genetic and environmental signals. And for this, I'm going to show you the example of a study that we have done in pulmonary arterial hypertension, which is essentially a disease of the pulmonary arteries, where in the patients, you have this huge proliferation of smooth muscle cells, and then the volume of your arteries shrink dramatically, So you end up getting strokes and all kinds of bad things. And the only treatment at the moment is actually lung transplantation. In the healthy individuals, you have typically one layer of endothelial cells, one layer of smooth muscle cells, and then comes the connective tissue. So you have a nice volume for your blood to flow through. So we have gotten now cells or data from cells of this very inner layer of the endothelium of these pulmonary arteries from PH patients and healthy patients. And we have done chromatin marks as histone acylation, which is an active mark. We have 3D interactions, and we have RNA-seq. And the biggest surprise, and I should mention these biopsies have been cultured for 2 days, so they have kind of lost their immediate memory or their immediate environment from the disease versus the healthy state. So what was really interesting is we had 0 differentially expressed genes in these patients or in this data, but we found thousands of differentially active enhancers and also many of these differentially active TFs when we used the DiffTF that I just showed you before. And it also seemed that the transcription factors actually recapitulated very much what is known about the disease. So we went on generating these gene regulatory network models using transcription factors, regulatory elements, and the genes. And we then looked at what is the enrichment of what is the function of the genes that are now upregulated by these transcription factors up in the disease patient, in the patients versus the controls. And we found interesting terms. Among others, we found smooth muscle cell differentiation, which is interesting in the disease etiology, right? Because we find this proliferation of smooth muscle cells in the patients. But another thing that caught our eye was proliferation in response to TGF-beta. So we thought, okay, maybe these cells respond in a wrong way to a TGF-beta stimuli, which is one of these endogenous stimuli that you can get in endothelial cells. So what we thought is let's try to actually see whether if we stimulate now these cells and we specifically measure now the expression of the genes that are differentially active in their enhancers, that are connected to the genes. We stimulate the cells with TGF-beta. What do we see? And indeed, what we find actually is that when we stimulate with TGF-beta, in the control cells, nothing's happening to these genes. And in the PH cells that have already a little bit of an enhancer opening here, the genes start responding and really start differential expression. And this was now very interesting because the genes that mostly responded in this way, actually many of them were kind of this endothelial to mesenchymal fate transition. So we have now this hypothesis that these cells, these endothelial cells, even though they are still endothelial cells, they're somehow either primed to transdifferentiate, or they're primed to kind of give some signals to their surrounding smooth muscle cells to proliferate. So this was a very interesting kind of study, independent now of the disease, to see that the same environmental signal in the same cell types, but within different patients that on their own don't have any change in differential in gene expression. When we look at the different chromatin potential, we can potentially have very different outcome given the same environmental signal. So that's why I think now with our framework, we actually do have this opportunity to really connect the cell state, which is what I think is current, what I refer to as what is currently expressed in the cell, for example, on RNA level, with the cell's potential, which is how the cell can actually respond to a certain stimulus, like in our case, it was TGF-beta. So this brings me back, to our big vision statement. And what I've shown you so far is, how we can build these networks and how we can, to some extent, use them to predict a response in PH patients. And what I want to go now, and I've shown you how we can now integrate genetics, because we can have, genetic variants here, and we can have environmental simulations that affect the signaling. But what I want to go now in the next part is to look a little bit more into this part, to see whether we can now actually integrate the genetics. I actually haven't shown you much of the genetics yet. So this is what I'm going to show you in the next example. And one of the nice kind of biological processes that really integrates a lot of different layers is aging, right? Because it's really the interplay of epigenetics, because you have been living for a very long time, also signaling because there's immediate things happening, and genetics, of course, because that's always what you have in your genome. So the system that I'm going to introduce now is actually an aging model that we have in our group. And this is the bone marrow niche, and we're specifically interested in these mesenchymal stroma cells. And the reason why we think that these are very interesting cells is, first of all, they actually are the precursors of all the the mesenchymal stems or the stroma cells in the bone marrow niche. So these are the adipocytes and also osteoblasts. So they're kind of the precursors of the bone, but also fat cells. And they're very, very lowly abundant. So there's only about 0.1% of the bone marrow niche actually is these cell types. And they are very heavily interacting with the blood cells, specifically with the hematopoietic stem cells, which again is a very small proportion. It's about 1%, which then make up the whole blood, and 99% of the bone marrow is indeed blood cells. So there's a lot of these interactions, and there's also the cells differentiate. So we think this is a very interesting model to study aging, because there's a lot of changes that happen to your immune system actually in aging. So since these cells are very rare, what we're doing is actually we're taking bone marrow stroma cells from donors, from healthy donors. We culture them in a dish to expand them, and then we do different assays. We do chromatin accessibility, transcriptomics, and proteomics assays. And our donors range between 20 to 70 years. So we really have a nice range of ages in our cohort. So just to give you again a kind of a big picture on kind of why I think multi-omics is a very important kind of strategy to understand like complex processes like aging, is when we look, for example, at the proteomics level, which is what you could say is maybe the most functional most functional part of transcription factors, you observe, most functional part of the cell, you observe that, of course, there's only about half of the whole genome that's actually covered, detectable by proteins, whereas by RNA, we can detect almost twice as much, and that they are toxic, and the RNA, like the toxic in the promoter accessibility, this is very similar to what we can detect on the RNA sequence. And what's important, I think this is what I showed you before, exactly these pictures, is that transcription factors are actually highly underrepresented in the proteomics level. And I'm just showing you here again this concept, that we can now use the chromatin activity that I showed you before, the chromatin accessibility, as a readout of this transcription factor activity, which is kind of the functional part that we're missing from the proteomics part. Okay, enough of the concepts. Let's just see how aging now affects each of these layers. And what was very interesting to observe is when we look at all the age-sensitive genes, so these are genes that are age-sensitive at any one of these layers, they tend to not overlap very much. So they either stay age-sensitive on one or the other layers. Interestingly, we find again nothing on the RNA level. So this is very much similar to what we saw in the PH project, and it may again really be because these cells are cultured, so they may have lost their kind of immediate functional environment. What is very interesting when we look at the chromatin level is actually they often fall in these bivalent regions, and all these bivalent regions seem to close with aging. And I'm saying this is interesting because bivalent regions tend to be the regions that are open in stem cells that can kind of get closed upon differentiation. So what we think this is kind of highlighting is kind of a loss of stem cell potential potentially in these aging cells. So we lose these regions that become open upon differentiation, which means that we kind of lose differentiation potential. What is interesting on the protein level, and I'm just showing you here one glimpse of it, is that many of the chromatin-related proteins, and here I'm talking about chromatin modifiers, chromatin modification enzymes, or also PRC complexes, they tend to be all downregulated in aging. So this could explain actually that why we have such a phenotype on chromatin, because the proteins seem all to be downregulated somehow. The TF activity was the most interesting part actually, because here what we found is a very strong difference between the old and the young in the terms of the transcription factors that are involved in adipogenesis, so the precursor of fat cells, and osteogenesis, and the precursor of the bone cells. And this is physiologically actually has been observed since the 70s, that as we age, our fat mass in the bone marrow increases, whereas our bone mass decreases. So this is a kind of related, it's a kind of explaining, or like very nicely aligned with a very physiological finding, or very physiological knowledge of the actual bone marrow niche. So we find old people MSCs from old people tend to become more adipocytes, whereas from young people, they tend to become more osteoblasts. So we wanted to understand a little bit more about how this is, how this differentiation priming may happen. And for this, we took a data set from Susan Mandrop's lab that she published in 2019. This is a great data set, again, multi-omics, RNA-seq, DNA-seq, which is essentially chromatin accessibility and also some ChIP-seq. And we could build a gene regulatory network based on this data set that is essentially then kind of reflecting the process from MSCs to adipocytes and to osteoblasts. So this is just some of the statistics of the network. So we have about 3,000 connections, 160 TFs, and so on. And what is now very interesting, if we now again do a very similar analysis that we had done with the pH, and we just take the transcription factors that are old in the young, we find that they tend to be connected to genes that are involved in adipogenesis. The ones in the old, they tend to be connected to genes that are upregulated in just the mesenchymal stem cells or stroma cells, and not so much the osteoblasts, but really the stem cells still, whereas in the old one, they tend to have this shift already in the adipocytes. So this is kind of getting a bit to the molecular understanding of what this could mean, or how this priming could be happening. And now, of course, we wanted to look for specific SNPs, right? Because as you know, aging is essentially a big risk factor for many common diseases. So we thought, now we have this nice framework. We find these age-sensitive chromatin marks, and now we wanted to understand whether some of these single nucleotide polymorphisms that are associated with diseases could be enriched in our MSCs. So this is essentially the question, how does aging contribute to the complex traits and diseases? And what was very interesting, actually, when we just did an enrichment study trying to understand which of the traits had SNPs that are enriched in any of in our aging genes, either transcription factors, proteins, or in the promoter, we find quite a lot of kind of bone-related traits, such as body height, bone mineral density, and also BMI, which is related to your fat content as well. Then, interestingly, we find a lot of immune system related traits, right? And this is interesting in the sense that MSCs are involved in the niche, right? And they can regulate these hematopoietic stem cells. So this was a very interesting finding. Then we also found some other traits that kind of, if you Google it, you can always find a link to MSCs, but I'm not going to go into it. So these are the age-sensitive genes, and what is interesting now is that even if we look at the expression level now of these genes, we group them essentially by expression level in the blood versus our bone marrow stroma cells, and even the ones that are much more higher expressed in BMSCs, we still have a couple of genes that are actually associated with these immune system traits. So this to me really indicates that these genes in the MSCs could somehow influence immune system traits. So we went back to our GRN and we were asking two questions. We were asking first, do we find, what are the genes that we find, is there some traits that we find actually enriched among enhancers that are regulating genes that are upregulated in the MSCs or upregulated in the adipocytes? And here we find some interesting associations. The most interesting one, I think, or the most easy to explain one is this adipose tissue, that we find up in the adipocytes and downregulated in the PMSC, so indicating that this is kind of the pathway maybe that these enhancers are enriching for. But then what I find more interesting is we then actually looked at which of these SNPs are regulated by age-sensitive transcription factor. So what that means is that you would have a SNP, and by aging, you would kind of start to enhance this effect that the SNP would have on the disease, because the transcription factor would get more active or would get less active. And here we find now, some traits that are related to physiological bone marrow aging, with the more fat and the less bone, right? So this is again the visceral adipose tissue that we find up in the young, and BMI that we find down in the young, and the bone density that we find up in the young. So it is really kind of reflecting again this physiological aging, indicating that indeed we find something that's physiologically relevant. So another one that I thought is actually very interesting is this asthma trait that we find upregulated in the young. And allergic asthma is actually something that has been associated more with young age. And there is indeed a lot of clinical trials that are now starting to treat asthma with MSCs, mostly for their anti-inflammatory role in remodeling immune cells. So I think what we have found here is a potential way kind of understanding, now we have our gene regulatory network, we have the transcription factors, we can now understand what are the genes that may mediate this effect of aging and asthma related or immune modulation. And so this is the end of my aging study. And I just want, before I stop, I just want to give you one kind of thought experiment, or like something to think about, which is completely unrelated to what I've been talking about now in the aging, but it is related to variation. And it is related to a study that actually a PhD student in the group has done, which is to try to understand whether there's anything that can explain variation across individuals in terms of gene expression. And of course, I wouldn't tell you this story if we didn't find anything. We actually did a random forest model, trying to predict the kind of variation across gene expression, both in a very controlled setting in Drosophila and one across human tissues, very uncontrolled setting. And we are able to predict variation across individuals with a really good R-squared of about 0.04. And what was very interesting is that this variation across individuals was equally predictive actually for differentiation across experimental conditions, let's say, treatment versus non-treatment and so on. So essentially we found features, and the features were mostly related actually to promoter complexity. So the more complex the gene was, the more stable it was expressed. Whereas the most simple one, let's say it had only one transcription factor binding site, for example, it was very variable. And if you think about it, it makes a lot of sense, because the complex regulation probably has a lot of buffering. So in each condition, there's a transcription factor that can activate that gene, whereas in a simple regulatory setting, you would only have in one condition the transcription factor that can activate it, whereas in all other conditions, you don't have this factor. So now I'm coming to what I told you, that is this maybe only a very small subset of genes that actually integrate these two signals that I've been talking about my whole talk about, these environmental and genetic signals? Is it maybe just these variable genes that seem to be responding to genetics, but also to environment? And there's some hint to this that we found that the drug targets, which I read as, genes that if you change their expression, they have an effect on the phenotype, right? Because otherwise they wouldn't be drug targets. So they tend to be the ones that are highly variable. So they tend to be the ones that can maybe integrate these environmental signals with your genetic and your genetic signals to actually cause a phenotypic difference. So this is what I wanted to tell you. And just to briefly get to my take-home message and my summary. So I've shown you that we have developed this framework of gene regulatory network models that integrate the signaling through transcription factors, regulatory elements, and the genes to give us a way of trying to integrate these genetic and environmental factors using the system epigenetics approach, to look at this context-specific signaling towards getting mechanistic insights. And I've shown you two specific examples. One is this enhancer-mediated priming that we find as a disease mechanism in PAH, and the other one is when we looked at the mechanisms in the aging of the bone marrow niche. And with that, I would like to thank a lot of people in my group. I mostly have to thank actually Christian and Ivan for developing a lot of these tools. The DiffTF, for example, was from Ivan and Christian. The gene regulatory network was developed by Christian and Ariane. Olga was doing this last part that I've shown you, and Brian was driving the aging related part. Armando, who's a postdoc, already left, was driving the pulmonary arterial hypertension part, which was actually a nice collaboration also with our collaborators in the clinic in Stanford. And then there's a lot of other people I need to thank for many other projects and contributions. And with that, I'm happy to have a discussion if you have any questions regarding these topics.
Thank you, Judith, for a really amazing, awesome talk. I will start. I have a couple of questions of my own, but I will start with some that we had. I think there was on YouTube. Some people who were watching. Just give me a second. So it was Sakib from Göttingen. He has a question regarding the first example you showed of pulmonary arterial hypertension, and he's asking, was the ChIP-seq, RNA-seq, and Hi-C done from the same samples? And if not, is this a potential problem, and how to circumvent it?
Yes, it was done from the same samples. I mean, we grew the cell. No, the Hi-C actually not. The Hi-C, we pooled everything together. And to get, because with the Hi-C, I don't think you get patient-specific differences. Hi-C has such a bad resolution at the moment. Actually, it was not Hi-C, it was CHIA-PET we used. We used CTCF, cohesin-mediated CHIA-PET. And that was done with all the samples pulled together. So we just wanted to get an idea of what are the loops, because we don't expect the loops to change so much. And for the RNA and the histone marks, these were done on the same samples, yes.
Thank you. I have kind of a related question. So if I understood correctly, your package to estimate or to infer gene regulatory networks is not taking into account SNPs or individual variances. So it's working still on the level of population. Is this something that you thought about doing? Is there a power to do?
Yes, I mean, we have done, of course, this analysis. So I think it actually takes information from SNPs into account, right? So I think what we find, I haven't shown you this, but if we look at our gene regulatory networks and we look at the links that we get from enhancer to gene and we overlap that with EQTLs, we get a super high enrichment, right? So we find a lot of the enhancers that we find are also EQTLs. But the thing is, I think we find much more than the EQTLs. And I think that's one reason is that EQTLs, of course, you have to do them, exactly in the right tissue so that we can compare it. But also, I think there is to some extent, probably some epigenetic variation across individuals, right? And that you wouldn't capture with the genetics, but you can capture it with the chromatin marks. So I think the chromatin marks capture the SNPs. Usually they capture the SNP information, but they capture also this epigenetic or kind of on top of genetic signal as well.
Okay, awesome. Thank you. I think we don't have any more questions. Let me double check once again. Yep, so with this, we thank Judith once again. And if somebody has, or somebody comes up with more additional questions, something, I'm sure they can reach out to you via your email or other contact. And with that, we would like to close this first lecture. We will have, I think, a break before the next talk in our series of seminars, and this will be talk by Izabela Makalowska on the retrogenes starting at 3 o'clock. So that will make some 15 minutes break, and happy to see you again on the Zoom. Just take note that it's a different Zoom link, and you can find it on Discord.
Okay, thank you very much.
Thank you again.
And reach out if you have any questions. Bye-bye.